Hey there! Welcome to this Smallmetrics video on DNA. DNA is the initialism for deoxyribonucleic acid. DNA is the organic chemical of complex molecular structure located in all prokaryotic and eukaryotic cells, as well as in many viruses. DNA is what makes up the codes for genetic information of inherited traits to be transmitted. The way DNA is structured determines things like the hair length of a dog, the color of your eyes, facial structure, and even things like the effect of a virus on its host. In 1953, James Watson and Francis Crick discerned the double helix structure of DNA. The double helix consists of two DNA strands wound or twisted around each other in a sort of arrangement that looks like a spiraling staircase. Each strand is made up of a long chain of monomer nucleotides. Nucleotides are the building blocks of nucleic acids, DNA and RNA. The nucleotide of DNA is made up of a deoxyribose sugar molecule, or a 5-carbon sugar molecule, and attached to this sugar molecule is a phosphate group along with one of 4 nitrogenous bases. The bases consist of 2 purines, adenine and guanine, and 2 pyrimidines, cytosine and thymine. Nucleotides are bound together by covalent bonds between the phosphate of one nucleotide and the sugar molecule of the next nucleotide. This bond forms a phosphate sugar foundation from where the nitrogenous bases project. The strands are held together by a hydrogen bond between the bases. Sequencing of this bonding is very particular. The purine adenine bonds only with the pyrimidine thymine, and the pyrimidine cytosine bonds only with the purine guanine. Because of this highly stable configuration of the DNA molecule, it allows the DNA molecule to act as a template for the replication of a new DNA molecule, but also for the transcription of the related RNA molecule. A segment of DNA that codes for cell synthesis of a particular protein is called a gene. So, the way the DNA replicates is by separating back into 2 single strands. These 2 now separated strands are what act as the template for the replication to take place. The new strands are copied by the same principle of hydrogen bond pairing between bases that make up the double helix. Once the 2 strands break apart and replicate by bonding to another strand, each new DNA molecule contains 1 strand of the original. This is often referred to as semi-conservative replication and is key to the stable inheritance of genetic traits. Within a cell, DNA ordered into dense protein DNA complexes are called chromosomes. Inside of eukaryotes, you can locate the chromosomes inside the nucleus. However, DNA can also be found in mitochondria and chloroplasts. Nearly every cell in a person's body has the same DNA. In prokaryotes, all the DNA is a single round-shaped chromosome in the cytoplasm. There are a few prokaryotes, bacteria for instance, and a few eukaryotes that have extra chromosomal DNA called plasmids. Plasmids are self-replicating genetic material. It may be helpful to think of DNA molecules as individual instructions that our body uses to make proteins and molecules essential for growth, development, and health. Thanks for watching this video over DNA. We know that studying for a test can be stressful, so be sure to check out our other videos to help you feel prepared. Until next time.